So the brain is very complex, so I don't think this is news to anyone. It has, for just to throw some stats out, it has 100 billion cells, and all these cells make connections with other cells. And on average, there's about 10,000 connections that one cell will make with other cells. So you can imagine this is just, it's more complex than the internet, which makes it incredibly interesting to study, but also very difficult. And that's kind of why I got into the field I'm in, because I always thought studying the brain was just absolutely fascinating. And whilst I'm trying to learn how the brain grows, and I'm mainly interested in how this grows in the embryo, so from a very small number of starting cells up to this 100 billion cell juggernaut of all these different connections, how does it actually form? And one way that we can look into this is to perhaps have a look at instances when this has kind of gone wrong. So when we have children born with problems, it's kind of like a window where we can say, okay, well, this child is actually having a problem with the way their brain functions, therefore it's probably the way that it's grown it's gone a little bit wrong. So we can find out perhaps what genes might be altered in this child. Those genes by default are involved with brain development. And that's where I really struck gold, because I did a PhD looking at how the brain grows, and then I married my skills with a guy called Joseph Getz, and he's a world-class geneticist. And this is what he does. He finds the problems in genes, looking through the genome. And so, these are, intellectual disability. We probably all know a child or a family that's been somehow influenced by a child with these sort of disorders, and here's just some of the more common ones. But it's actually probably more prevalent than you think, and that's because the children that are affected, and then it's not just childhood, these children turn into adults, and quite often if you have an intellectual disability, it doesn't really mean you're going to have a shorter lifespan, it just means you're going to need care for your whole life. And so whilst it affects actually 2 to 3% of the world population, we probably don't see these people in our everyday lives just because they're secluded away, just getting the help they need to sort of get through life. But it's a huge problem. In terms of economic burden, it actually costs more to deal and to care and treat for these individuals than it does for all cancers combined worldwide. And that's because it is a lifelong treatment, whereas cancer is more of an acute thing. You do the treatment, you heal or you perish. So in Western worlds, there are causes of intellectual disability that we can control. So fetal alcohol syndrome, or even in third world countries where they don't have enough iodine, this means that the children are born with deprived of nutrients, that are deprived when they're growing during pregnancy, and they come out with problems. But particularly in Western worlds, we can control these environmental things, control infections, put out plans to give the mothers the healthiest possible pregnancy, and we can eliminate a lot of these environmental factors. But even with that removed, 50% of all children that are born with these neurodevelopmental disorders arise because they have an alteration in their genes. We all have alterations in our genes. Each one of us in the room, we have 25,000 genes, and each one of us, on average, has about 20 genes which are completely non-functional. And we have 2 copies of each gene, and I'm talking both copies, so we're all carrying complete gene knockouts. We're all here standing, we're all warm, we're probably all reasonably highly functioning because we're here listening to science. The genes that we have knockouts are redundant genes, they're backups, other genes that can do the function. But for these people, you get the wrong mutation in the wrong spot, and you can have a real problem. And so finding these genes that have these alterations in these kids is really, as I mentioned before, the window into sort of understanding which genes are controlling brain development. And because they're genetic, we're always going to have this problem. And you can wipe out all these environmental causes, but we're always going to have these problems in DNA. So this is just it's going to be an ongoing problem for society to deal with. And as I mentioned, the economic burden on the health system, and particularly on the families that have these children in them, is enormous. Okay, so this is kind of like the set out of the lab. As I mentioned, we start off with basically the research begins and ends with patients, which is a good place to do it. So we you have cases where often there's a family history of a disorder, say epilepsy, your uncle had it and your brother had it, so perhaps it's going through the family. And in this instances, we kind of feel like there's a good reason for the genes that will be involved. And as I mentioned, the lab's really well set up to have a look at these genetics and try and find the genes involved. If we can find, you've got a mutation in gene X, that's really good to relay back to the family because there is perhaps a sister who's unaffected and she wants to have her own family, but she's unsure. She's unsure if she's going to pass on to her children this gene mutation. So passing this information back to the family is really valuable, but it doesn't necessarily help the affected son. So that's kind of where it gets a lot more trickier. That's where we need to kind of work out what the gene does, and that's kind of like my real passion in the field, in this diagram, I guess, is to try and work out how genes control the way that brain cells grow, how they connect with one another and how they function to communicate properly. Because it's only with this knowledge then we can begin to really make some quite personalised or targeted therapeutic interventions. And this can be as easy as changing diet, but often now it's much more complex. But we need to do this functional study to really get an insight into it. And as I mentioned, the lab's mainly interested in these neurodevelopmental disorders, intellectual disability, epilepsy, and more recently in cerebral palsy. So, I guess I'll break talking about this genetics, and I'll just digress a little bit for some interesting conversation, perhaps, and then I'll look into how we're actually using stem cells to do some of this functional work. So the genetics. This is really about reading the DNA. The DNA is just a massive long chain, I guess, for an analogy, of 4 different types of links, A's, T's, G's and C's. These are the bases of DNA. And they just form this really long chain, and it has 6 billion links in it. And so when we get a patient with an intellectual disability and we want to find out which gene is involved, what we're doing now is actually just reading the entire genome. It's one base pair at a time through a whole 6 billion bases. And within this sequence, there'll be genes, and the genes are the parts of the DNA we know most about. And if we find a change in a gene, then quite often that begins us on a journey to try and work out what that gene does. Does it make sense that a mutation in this gene causes intellectual disability? The gene, for example, might be something that controls toenail growth. It's probably not going to be involved. Or it can be something like a gene that controls chemical messaging across the synapses in between where neurons communicate, perhaps more likely. But we need to go and investigate exactly what these genes do to really get some confidence one way or the other. So, reading this DNA is kind of like there are 7 billion people in the world, right? And we're looking at 6 billion base pairs to read through. Sometimes the cause can be quite massive. For Down syndromes, it's an extra whole chromosome. For other genes I've discovered and worked on, it can be a one single base pair change. So it really is just kind of like playing a massive game of Where's Wally, global wide. And the only way we can do that nowadays, we used to do it kind of like by looking at candidate genes. So we used to look at what genes have been involved in the past. Maybe the clinical features of this patient, which is consistent with another patient we've seen before. We know that patient had a problem with this gene. So we used to go one by one, gene by gene, trying to find out what was wrong. But now we just look at the whole genome. Sometimes just the bits that code genes, but then sometimes the whole lot. And this is just because it's basically a technological advancement in the last probably 5-10 years. And to explain this, I guess we have a look at the very first genome that was ever recorded. It was published in the year 2000. It took 13 years to count with the old technology. It took 13 years to gather all the information to work out, base by base, what the DNA chain looked like. And it cost $3 billion. Today, I can send off, I can just take some blood from a patient, extract the DNA and send it off. It'll cost me about $1,000 and it takes 2 days. So this is a massive change in the way that we can approach our research. but it's also going to affect us all in the way that we, I guess, we interact with healthcare. And I'll get to this in a second. I guess just using this technology with me and myself, I've been involved in the last 5 years discovering 8 new genes that are involved with epilepsy or intellectual disability, but the lab itself, the larger lab, has been involved with over 50. So it's really making some groundbreaking discoveries. And as I mentioned, this information back to the family is essential for them to go and have confidence in having more children. So, I mean, this technology is really at a rapid pace advanced how we can discover new causes of genetic disease. So it's not just neurodevelopmental disorders, but any sort of genetic disease. The ability to sequence DNA rapidly and identify these changes has just changed the way that research has been done. Another example is cancer research, which is a disease where cells they lose their ability to fight against new mutations, and in fact they're indeed with the ability to kind of hypermutate. And understanding which mutations in cancers are really driving tumour growth is something that's really being promoted and benefited by these new sequencing technologies. But as I mentioned, now because the technology is so freely accessible and cheap, it's actually now being used straight as a diagnostic test. So if you go to Nijmegen, as I mentioned, we used to go gene by gene when we had a child with epilepsy. Now in the Netherlands, in a facility I visited recently, every child that has some sort of problem just gets their genome sequenced. And then it's like, okay, well, you get the information that you need, you perhaps find a change in the gene that makes sense. You've got epilepsy, and that's because, yes, you've got this mutation in the gene that we've seen in hundreds of other patients, so it's most likely that this is the cause. And then that can lead to kind of particular drugs that you give a child. You can go back to see what the other children, what was effective and what wasn't, and really give a kind of precision medicine. But it also means, because you've looked at all the genes, that you have what we call incidental findings. So, that explains the epilepsy. We can treat that, that's fine. But by the way, what we also found was that when we looked at your BRCA1 gene, which is the gene involved with breast cancer, you've got this change that probably means that you might be a little bit worried in 20 years' time that you might get breast cancer, or that you've got some genetic changes that actually predispose you to Alzheimer's or Parkinson's disease later in life. And this is now what we call these incidental findings is really, I guess, a challenge now for genetic counselling. So we can get the information, but what do we tell the patients? What do they want to know? So do we tell the patients, okay, you've got a chance of getting Alzheimer's disease, do you want to know this? And because if you tell them, then they can perhaps step in there and change their lifestyle and maybe make their lives, kind of benefit them and protect them against the disease. But at the same time, you put them in a lifelong burden knowing that everything they do, in the end, they're going to get Alzheimer's or Parkinson's disease, and it's going to be really hurtful. So, I mean, these are the questions with this new technology are throwing up. It's a new age of healthcare that in 10 years' time, a lot of us will have our DNA sequenced, a lot of our young children are, and it's just how we deal with this information. In some countries, in Iceland, for example, every child that's being born is being recruited into a cohort, and they're getting the whole DNA sequenced, and then they're going to track these children throughout their whole healthcare life over the next 20, 30, 40 years. And that way they can have a look at, on a population scale, what sort of mutations are maybe protected against certain diseases or infection, maybe what sort of changes in DNA are associated with, educational success or failure. And so the more and more people get their DNA sequenced, and the more they start to make these associations with how these changes in DNA affect their life, is going to be kind of part of the new healthcare, I guess, benchmark. So I guess we're just at the start of it now, and by the way, I'm not a genetics, I don't do much sequencing, this is just the one arm of the lab, and I see this communicated all the time at conferences, and I just think it's it's something that hopefully you guys will find interesting and keep an eye on as time goes on. By the way, Adelaide's actually been pretty active in this area, and they are the first, the IMBS in Adelaide are the first diagnostic service in all of Australia to actually have accreditation to do whole genome sequencing and to perform these sort of tests. And so it's not just, in certain parts of the world, it's happening right here in Adelaide right now. So that's the genetics, but the title mentions stem cells, so I will move on, because as I mentioned, I've discovered a few genes, and for me, I think the real exciting thing is to then try and work out what these genes actually do in terms of making the brain grow. And so the sort of resources that I have available to me are to use the cell lines from the patient, so we get their blood. That's good for us to get their DNA and start to have a look at what the changes might be. But it's not very good if I want to look at a blood cell from the patient and try and work out how their brain might not be wired properly. It's sort of the wrong material. So for this, there's a couple of things we can do. One thing that I have done previously is use small animal models. And currently in the lab I use mice, small rodents, or we also use zebrafish. And kind of like, here's the reason why. This is an embryo. One of these is a mouse and one of these is a human, and you can see that they look quite similar. At this embryonic stage of development, a lot of mammals kind of really do look very similar, and that's because from a very early stage, the blueprint for their embryology is very similar. And so whilst I'm looking at studying human embryo development, I can use the mouse and at these very early stages be confident that the changes, well, the readouts that I'm seeing in the mouse embryo are quite relevant. You can see this one here has a tail, so that's the mouse. And so as I mentioned, we can, say for example, I find a gene and no one knows anything about this gene. One thing I can do is say, well, where is this gene active in the mouse? And here's a gene, it's called USP9X, it's one that I've worked on. And in purple, you can see in the purple areas where this gene is actually active. And it makes perfect sense that you can see it's quite active in the brain. So this is a good candidate to chase up on. We can manipulate the mouse fairly easily, and it's becoming more and more easier, I'll admit. With some new technology also. And for this gene, we found mutations actually, we hypothesised that would make the activity of this gene less. So what we did is we made a mouse that didn't have this gene working properly. And we can have a look at their brains, and just have a looking down this part of the brain here called the hippocampus, this sort of jelly roll structure. You can see in a normal mouse, it's just looking at the size. I just want you to have a look at the size of this structure versus that structure, and you can see that it's a lot smaller. Without going into too much detail, this part of the brain is essential for learning and memory tasks, actually forming new learning and memory tasks. And so even in the mouse, when we disrupt this gene, we start to see differences in the brain that kind of make sense with the phenotype of intellectual disability. So it's difficult working with embryos though. Here's just a size scale of a mid-gestation embryo. It's actually inside, it's a mouse mother, you pull it out, and it's just a fraction of the size of a 5-cent coin. So it's very hard to monitor what's going on in these animals. It's very hard to take material out to study it because it's just so limiting and so inaccessible. It's also very expensive, and to work on this mouse took me my whole PhD. So that's just one person working on one gene for 3, 4, 5 years. We needed a more high throughput method, particularly now when the sequencing is meaning the diagnostics, well, you're discovering new genes which you think might be involved at a much more rapid rate. So there's now over 1,000 genes implicated in intellectual disability. To make a mouse model and study each mouse is kind of unfeasible, although it is being attempted through large consortia. So another way to approach this is to use sort of a cell culture model. And these are growing cells in a dish. And I guess The cells in the dish are a reasonable model, but I sort of took an approach which was relatively novel, and that was to actually sort of do a hybrid where we take some cells out of the growing embryos and put them in the dish. And the reason we do that is because if you take the cells out of the embryo and put them in the dish, they still kind of, I guess, retain some of their origins. So they go on to behave in the dish as they would normally in the embryo, and that way you can monitor developmental processes that would normally be happening inside an animal, actually inside the dish. And because they're in the dish, that means they're completely accessible. Now I can manipulate gene expression much more easily. I can throw on different drugs, I can do different manipulations to try and push the culture one way or the other, and try a lot more high throughput ideas, I guess. So one cell culture model that I really enjoy doing, and there's a few of them, was to use embryonic stem cells. So here's just a progression of mouse embryo development. At 3 days past fertilisation, you have this sort of stage called the blastocyst, and this hasn't even been implanted into the wall yet. It's just but it does contain a cell population called embryonic stem cells, and those cells can actually give rise to every cell type in your body. And this is for mouse, but it's the same in human. They worked out quite some time ago that you could actually take these cells and put them in a dish, and under certain culturing conditions, hold them in this sort of blank, unpatterned stage. And then with the right conditions, you can direct them to turn into different cell types. So for me, having these cells in the dish meant I could alter the genes, and I always looked at manipulating genes which I thought were involved with neurodevelopmental disorders, and then have a look at how the neurons that end up being produced behave. I also looked at getting cells from different stages of mouse, and I guess the two stages which I wanted to make a highlight of are that you go from an embryonic stem cell, so these cells can turn into any cell in the body. They end up giving rise to a second sort of stem cell, which is specific for the brain. So then you go from a cell type, these are restricted, these neural stem cells, in that they can't give rise to any cell in the body anymore, but they can give rise to any cell type in the brain. And then the brain, these neural stem cells, they sort of divide, and that's what makes the embryo grow, makes the brain grow, and then in the end they all differentiate, or they all divide to give rise to a cell that doesn't divide anymore, and that's your neurons. So when they say, you're born with the neurons that you have the neurons that you were born with, that's mainly because your brain is full of all these cells that don't divide anymore. All this cell division and growth occurs during embryonic development, and very little of it occurs in the adult brain. So if you take, so here are these stem cells again in just a cartoon form, they form the second neural stem cell population, and these actually arrange themselves into a tube. And that's pretty much what your brain is. It is, if you kind of boil it down into its most simplistic cartoon picture, it develops as a tube, and it remains as a tube even in adulthood. The cells sort of radiate outwards, kind of like spokes on a wheel, and they join all together in the middle, and that makes kind of like a channel in the middle, and that's where your cerebral spinal fluid sort of washes around. And so your spine in the brain is just one big long continuous tube. So during development, these stem cells' role is to first of all divide, right? So you can, so the embryo grows, and so you can get a base number of cells, and then each stem cell sort of divides into two and produces two neurons, and that's it. They don't divide anymore. So there's your neuron, your progenitor cell in red, dividing into two neurons, and they're the neurons that you'll have for the rest of your life. And this is a process called neurogenesis. And these neurons sort of, in the embryo, they're born near the central bit, and they migrate outwards. So can we recapitulate this complex process in the dish? Well, here's what it looks like when I'm using embryonic stem cells and looking at this gene USP9X. So you can see the similarity between this structure here, the neurons around the outside, the progenitor cells sort of radiating in the middle, and this central ring of cell adhesion. You can see that perfectly here in the dish. So this is just like a cross-section of a neural tube. But this is embryonic stem cells which are asked to turn into neural tissue growing in a dish. So you can see the similarities. And embryonic stem cells have a fascinating ability to self-organise. So you can put these sort of cells into the right conditions and grow structures that look like eyes, or you can grow structures that look like the highly architecturally sort of fascinating kidney lobules, and all different types of sort of basic fundamental units of the human body. You can sort of ask stem cells to do this in culture. And so here's a gene which, when it's this USP1X gene which I mentioned before, so this is when it's sort of working normally, you get this nice structure. And when you reduce the activity of this gene, what you see is you lose this architecture. So that, again, this is telling you, like before, that I mentioned before in the mouse, that this gene was important for hippocampal development. Well, here it's basically the data is telling me that this gene is also important for the very earliest stages of neural tube development, because when we knock out the gene, we lose the architecture. And we also kind of can see this in the mouse, which I'm not showing you here. So that's not all. I guess we can model in the dish. So as I mentioned, so here's the progenitor cells. They divide to give neurons. These neurons sort of migrate outwards. And so about the end of embryogenesis, once all the progenitor cells are sort of depleted, you're just left with this network of neurons. And that's basically what we have throughout life. So we can study these neurons actually in the dish as well. So at the end of differentiating these embryonic stem cells into brain cells, we're now looking at how these neuron cells sort of make connections with other cells. And again, here's USP1X. Here's what a neuron looks like when it's sort of branching out to make the very first beginnings of these connections, and I mentioned each cell can make up to 10,000 of them. So here's the sort of the cell body, and it's sending out these processes to touch and communicate with other cells. And when we decrease the activity of this gene, you can see that the growth or the elaboration of this neuron is stunted. So this is kind of telling me that this gene is also important for the way that neurons can integrate into networks of neurons in the brain. And lastly, neurons aren't the only sort of cell type you have in your brain. There are also support cells, which are astrocytes and oligodendrocytes. These cells, like the oligodendrocytes, put kind of like like insulation on the nerve fibers so that transmission occurs quicker, and astrocytes provide protection against toxicity. And you can also study these in the dish as well. So if you push the differentiation system another way, you're not only just getting neurons, which are here in green, this is a neuron cell, but you're getting the astrocytes, which are coloured blue, and also the oligodendrocyte cells, which are coloured in red, and you can quantify the differences that your manipulation might make. So this is really studying processes of embryonic brain development in the dish. Okay, so what I just wanted to lastly touch on was kind of, I guess how we now move the model from mouse cells to human cells, and what the advantages of using human cells could mean. I mean, to this point, I've used the embryonic stem cells as a model of brain development, but there's much more potential. And a couple of recent, I guess, developments in the field I think are worth sharing because they're quite interesting. As I mentioned, I've talked about model disease, but you can imagine if we're just looking outside now of neurodevelopmental disorders, and perhaps say someone with a spinal cord injury, they're quadriplegic because some of the nerve cells in their spine are severed and they don't repair very well. The promise with stem cells was that we can make these new nerve cells in the dish, and we can inject them into your spine, and they can reconnect and restore your motor activity. And this has been a bit of a pipe dream for stem cell biology for a long time, but nothing's really ever come out of it. And there's been a few hurdles along the way, I guess, and that's mainly because the source of cells, I mean, we're dealing with human stem cells, so these are obviously derived from human embryos now. And the ethics and the stigmatism against using these sorts of cells and where they come from has really hindered the progress. There's been a few cell lines, and once you have these cell lines, you can grow them for years. Some of the human embryonic stem cell lines have been in culture for 20 years. You can keep them in this blank sort of stage and just tell them to turn to your cell type when you're ready. So there was a limited number of cell lines available. And of course, we're all different genetic backgrounds. It's just kind of like an organ transplantation. If it's not the right genetic makeup, then your body's likely to reject these cells as foreign, just like the problems with organ transplantation. So a few years ago, in 2008, a discovery was made by a Japanese group, and it's kind of really revolutionised the way that we can get over these hurdles. And that was an amazing discovery. Usually, he got the Nobel Prize for his discovery in 2012, which is 4 years after he made the initial discovery. Usually a Nobel Prize goes out to a discovery maybe 40, 50, even when 50 years later, when the impact is fully recognised. This happened in 4 years, to just, I guess, put it in perspective. And that was, he worked out a way of turning anyone's cells into embryonic stem cells. And he did it using skin cells. So we can take a skin cell from me, from anyone. It doesn't have to be skin, it can be blood. And he's got this method, which is very easy to use. We're using it in our lab also, and it's been used throughout the world all the time, to turn your cells back into embryonic stem cells. And of course, this means now that we can use your own cells to make your own therapeutic relevant cells. So for this spinal cord guy I was talking about, he doesn't have to worry about immune rejection or where these cells come from. We can just take his skin cells, reprogram them back into embryonic stem cells, turn those stem cells into a cell type for him, a spinal neuron, and give it back to him and see how it goes. This is being done for the first time, and of course, there's also an opportunity to edit or repair genetic problems as well. So the very first time this was done clinically was just recently, and that was with a retinal disorder. So they took the patient's cells, they had a genetic mutation, they took some skin cells, they looked at the DNA, they found the problem, they fixed it, and there's this new technology that enables you to edit DNA very rapidly now. So they fixed the genetic problem, then they grew, I think it was a retina or a cornea in a dish, implanted it back into the patient's eye and restored eyesight. Very first time this sort of, in a clinical setting, that this cell therapy has actually worked. And it's kind of, I think it's the beginning. There's been a lot of promise for stem cells, but this new way or new technology of actually generating your own stem cells is really revolutionary, and it's going to impact, again, precision and personal medicine going into the future. So I just wanted to end it there. I guess for us, we're really working on our functional data. We're not quite, we have one clinical trial in operation at the moment. We're not really